Thank you, Melissa. That was fantastic. So I want to now start the research day, and our first speaker is Wendy Gold from University of Sydney, and she is going to discuss multi-omics analysis for a search for biomarker for STXBP1. Wendy?
Thanks, Jim. Good morning, everyone. Sure, I knew it was going to be hard to speak after Melissa. Thanks, Jim. But you know, I was just thinking and reflecting when you were talking, Melissa, how important it is to hear the stories of what I call rare parents, your stories which fuel us to get us out of bed every morning and to keep us doing the work that we're doing in the lab. So thank you for sharing your story. Today, thanks so much for to both Charlene and Jim for allowing me this opportunity to present our biomarker project today. We thought about doing blood biomarkers. We're looking into blood biomarkers quite a few years ago. And today I just want to share the data that I have today, where we asked a very simple question: can the blood from STXs reveal the molecular mechanisms of STXBP1 deficiency? As Charlene mentioned in her opening talk yesterday, there are 17 disease-modifying therapies that are either entering clinical trials or in clinical trials. And this is a great inflection point for STXBP1 because now we've got these therapies that are rapidly moving towards the clinic. And so I just wanted to share with you our biomarker study today because I think this is a really important time to start thinking about biomarkers and how we can introduce them into clinical trials. They'll be really important to inform early response to treatment and also to understand the molecular consequences of disease-modifying therapies much before we can actually see them from a symptomatic perspective. Now, while a lot of the clinical trials, not only in STX, but other rare disorders as well, the clinical trial outcome measures are dominated by carer and parent reports. These are really, really important. However, molecular biomarkers can help us understand what's happening at the molecular level right before the symptoms actually occur. So biomarkers are because they can help us with diagnosis as well. They can help us predict and allow us to see the difference between disease, not disease, and severity as well. They also can be responsive, but also they can help us identify disease drivers as well and drug targets, and that's why we think blood biomarkers are so important. We took a different approach to biomarkers. Instead of looking at individual omics, instead of looking at just the genes, just the proteins, we took a holistic approach where we looked at the transcriptomic, the proteomics, the phosphoproteomics, and the metabolome as well, which has lipids in it as well, and looked at them all together in a coordinated approach. Initially, we did look at just them individually because there is some value in looking at how genes and proteins and metabolites change. But what this doesn't tell you is the overall coordinated approach, what's driving the disease. So speaking to my bioinformaticians, I'm not a bioinformatician, I'm a neurobiologist. They suggest that we take on a MOFA approach, which is a multi-omic factorial approach analysis, which looks at all these molecules together, not just individually. And what MOFA does is identify latent factors. Latent factors are these hidden biology that you wouldn't normally see if you look at omic data individually. And so basically to break it down, Lima asks what molecules are significantly dysregulated, whereas MOFA asks what is the coordinated and shared biology across all these domains, because as scientists we know that these molecules don't work individually, they work in a coordinated molecular mechanism in the cell together. So this is our study design. Thanks to the STXBP1 families in Australia, they helped me recruit 22 patients, 22 individuals with STXBP1, and we got 22 controls as well. Now, these controls were either siblings or a friend or a relative that didn't have STXBP1 or a known neurodevelopmental disorder. We took peripheral blood and we extracted the PBMCs and the plasma. From the PBMCs, we measured the transcriptomics, the proteomics, and the phosphoproteomics, and the plasma, we measured the metabolomics in an untargeted way. We then looked at the individual analysis, and then we integrated it into MOFA as well. So I'm just going to step you through the individual analysis first, and then MOFA, and then and I'll share with you our working hypothesis. Oh, what we also did as well, which is really valuable, and I hope that I have enough time to show this as I go through my slides, is that we had a participant survey. And what this participant survey did was help us to inform some of our understanding of the molecular analysis. So this was really powerful. So first of all, just to give you an overview of the spectrum of the variants, most of the variants in our cohort were, as we know, that there are the leading form of variants is a missense, and then we have had quite a few splice sites variants as well. We have one called TBC to be confirmed. We're not too sure about its molecular consequences, so we're still looking into that. This is a cartoon of all the variants that are nicely spread across the gene. And you can see by the functional domains and the exons that there is a spread of variants across the gene. And the lollipops just show where we had more than one individual with the same variant. The age distribution, unfortunately, we couldn't control for this. The STXs were a lot younger than our controls, but still in the pediatric range. STXs were 6.7, had a mean age of 6.75 years, and the controls were a bit older, they were 14. Gender distribution, we had a nice split, which was great to see for the STXs. However, with the controls, they were predominantly female. As far as the seizures go, 68% reported seizures in the STX cohort. As you can guess, there were none in the controls, which was good to see. So most of them did report seizures. Other illnesses, we caught this. We thought this was really good information to have, because it has allowed us to inform how we analyze some of the data, and I'll show you this in a minute. But with the other illnesses, there were almost 70% of individuals with STX that didn't have an illness in the 14 days prior to us doing the blood draw, and this is really important, and I'll tell you why. Sleeping, yeah, they reported that sleeping was not normal. And 70% were not sleeping normal to the age, and they woke up a lot. As far as medications go, we tried to capture as much as possible. We allowed them to enter free text as well with what medications they were taking, but predominantly, which was surprising, 54% were taking anti-seizure meds, and 41% were taking dietary supplements, and then there were some taking sleeping medication as well. And then we have a breakdown, but I don't have enough time to show you the breakdown of all the medications they were taking. So going back to our omic analysis, just to remind you of our study design, we took peripheral blood, we extracted the PBMCs, we extracted the plasma, and then we ran the transcriptomics, proteomics, phosphoproteomics, metabolomics from the plasma, looked at them individually, and then we condensed them together as well, and then looked at them together. This is the analysis, a PCA, principal component analysis of the transcriptomic data. As you can see, and you would expect, that the variance between the controls and the STXs separate really, really nicely. As far as the dysregulated genes go, we didn't see a lot of differentially dysregulated genes, although we did see some, as you can see from this volcano plot, that some had a really large log fold change, so the variance is on, the significance is on the Y and the log fold change is on the X. But we did see some interesting genes like MNDA and S100A9 that seemed to pop out really significantly, and also the fold change was high as well. As far as the proteomics go, we didn't see such a clear separation, and you can see that in the middle between the STX and the controls, there is an overlap there. This data we captured in the survey. The controls that are on the periphery, these controls had either asthma or an infection 14 days before, so they don't seem to cluster well with the other controls. And then in the middle, you can see that there are two controls that cluster with the STXs. Those are siblings. of their STXs. So they seem to align very close together. And there's one STX right in the middle at the bottom on 0. That STX is a very mild case. So they seem to overlap into the controls. Volcano plot with the proteins shows a nice array of dysregulated proteins with some highly dysregulated proteins, which I'm going to mention a bit later, for example, like Siglec-5 and also GRN. Now, what we wanted to do from this analysis was not particularly from the lemma analysis. Lemma is a bioinformatic term for dysregulated molecules, significantly dysregulated molecules. We wanted to look at the holistic approach. So instead of actually looking at individual genes and proteins at this point, we wanted to look at pathways. And what we found was this, and you'll see this throughout the rest of my presentation, was this activated monocyte state in the blood of patients with STX. So the monocyte differentiation maturation was genes that lead to monocyte differentiation were upregulated, phagocytosis, antigen processing, an innate immune defense response was upregulated, and protease control. So all the genes that were and proteins that were significantly upregulated were driving this monocyte and innate immune activation. The ones that were downregulated were genes and proteins that were involved in protein translation. So there was this coordinated approach of dampening translation. For example, mRNA, RNA were downregulated, and peptide chain elongation. So we had an upregulation of the innate immune system and a downregulation of translation. But what we wanted to do moving forward was to look at how we could coordinate this all together. So we looked at a MOFA, we took the MOFA approach, and what MOFA does is look at these latent factors that drive the biology across all the omics. And we found that with MOFA latent factor 1, which is the bottom line here, we could see that there was a lot of the variants, this is like a PCA plot, but it's in a heat map form, that the proteomics and the phosphoproteomics were driving all the variants. And we identified this is a volcano plot of the proteomics of the latent factors. We identified top 10 upregulated on the right with the red and top 10 downregulated molecules. And you can see here Siglec and GRN are some of the molecules that have come up again. And we found the same thing with MOFAs, that in the downregulated pathways, there was this lack of protein synthesis, a downregulation of translation. And upregulation, we saw again this innate immune system monocyte activation. We also looked at the phosphoproteome as well, and we saw that ApoBr was phosphorylated in 3 different places, and this was significantly upregulated. We're not too sure what this means yet. This molecule extracts lipids from the circulation, so we're not too sure what this means. Once we do the metabolomic analysis, we'll be able to understand this more. But if anyone knows what relevance this does have, we'll be happy to chat to you about it. What we did see from the phosphoproteomic data as well was that there was also this increased in innate immune signaling and monocyte activation. So it was concordant across the genes, the proteins, and also the phosphoproteome as well. And again, the downregulated proteins were this, showing that there was ribosome protein synthesis, downregulated, again, translation dampening. The transcriptomic latent factors, again, we saw NMDA and S100A9, which shows us this activation of immune response. Again, overall activation of immune response, downregulation of translation as well, so it's across all the domains. So, Jim, I'm almost finished. Proposed panel of biomarkers from all the molecules that we observed that were significantly dysregulated across the individual omics, across the individual transcriptomics, the proteomics, the phosphoproteomics, and then putting them all together into MOFA to understand this coordinated biology, we identified these 4 molecules, S100A, which is a calcium-binding protein, and it's one of the earliest indicators of systemic immune response. GRN, which is a progranulin, which is secreted growth factor. It's also highly expressed in microglia as well, so that could be telling us something. MNDA, which is a myeloid nuclear differentiation antigen, this primes, it's a transcription regulator, it primes the monocytes to be activated, and then Siglec-5, which is a break response, which is to try and regulate the immune response. And so these are central to disease biology because they show us that there's monocyte activation, a myeloid cell state change from NMDA, innate immune system activation, inflammatory signaling. We've got NF-kappa B and MAPK pathways that are dysregulated, and also a lysosomal neuroimmune biology response. And so these are easily measurable in blood, as we've shown, and there could be potential biomarkers. So our working hypothesis, this is just one of the working hypotheses that we've come up with at the moment. It may change when we get the metabolomic data, is you have STXBP1 deficiency, you have neuronal dysfunction, and you've got cellular stress in the brain. This leads to circulating markers, which the monocytes pick up, and they respond to this, and they become activated, and they form this activated state. And then we've got S100A, which turns on the inflammation, and MDA, which changes a monocyte state to the state of activation. We've got Siglec-1 saying, hey, don't overreact, let's try and calm down, and GRN as well to keep the homeostasis. But these are all upregulated, so there's no homeostasis in the blood cells. So our next steps, we think that 22 samples is a powerful number of samples, but I don't think powerful enough. I think if we have more samples in this cohort, that we'll get a clearer picture. So it'd be great to have more samples. We're also going to integrate the metabolomic data that STXBP1 collected, how long ago? 3 years ago? And put that data into our data and run it through MOFA2 as well, so that we have a large data set. What I think would also be really, really valuable, if we can identify these biomarkers in our mouse models, it would be really, really great if what we're looking for in the humans, we're actually seeing the same biomarkers in the mouse models. So when we're treating the mice with disease-modifying treatments, that we can actually analyze the same biomarkers that we will be doing in the humans. And then also what I think would be really, really valuable is to have a look at patients that have gone through clinical trials and identify those that have responded to treatment, those that haven't, and whether these biomarkers change accordingly as well. So we can stratify patients that have responded to treatment and not, and this will be a good validation to see whether our biomarkers are actually allowing us to understand whether they can pick up disease-modifying treatments in the blood. So in summary, we have answered our question. The blood samples can reveal molecular consequences of STXBP1 deficiency, and MOFA can too identify dysregulated processes, as I've shown, and disease signatures that can inform biomarker discovery. These are not necessarily the set biomarkers, because we've still got a lot more work to do. It can reveal disease presence. Will be really good if we can get clinical assessment scores of these patients, because then we can do a correlation between the biomarkers and the assessment scores to see if we can identify disease severity, because in the latent factors can then help us identify those that have low severity and those of high severity, and it can give us insights into the pathophysiology as well. So biomarkers such as blood biomarkers can help us identify an early response to treatment before you actually see that in the symptoms. It can correlate disease state and severity and enable early detection, which complements clinical and molecular diagnosis. So I'm going to end there and just acknowledge my lab. These are members of my lab and also my collaborators as well, especially Iggy and Nada. They did all the bioinformatics. As I said, I'm not a bioinformatician. And Fred and Tristan, who are at UQ and University of Queensland, who are doing the metabolomic analysis for us, and also to the STXBP1 family, especially to Lauren and Manjula, who lead the STXBP1 community and are really powerful advocates for the community, and they also helped us to collect these samples. My funding sources are below. We were fortunate to fund this project through the $1 Million Bike Ride. And then finally, this is a picture of Australian families. They all sent me pictures so that I can present today and represent them today as well. So thank you very much.
Have time for a question or two.
Yeah, I have one. Thank you. I guess I was hoping if you could elaborate a little bit more on just the connection between the brain biology and what you're seeing in the blood. Like what will actually happen if you change the levels of STXBP1 in the brain, and why would that be reflected in the way in the blood, I suppose?
Yeah, we've been scratching our head about this too. If we had a larger cohort, it would be nice to actually see the levels of STXBP1 protein and see if there is any correlation between this immune activation. I'm not too sure, but we've seen this in other disorders as well, like Alzheimer's and Parkinson's and other neurological disorders, where there is this pathophysiology in the brain, but there are these circulating biomarkers that reflect what's happening in the brain. So I'm hoping that we can have take learnings from the other neurological disorders like Alzheimer's to try and understand the connection between the brain and the blood.
Thanks.
Hi, my name is Sepita from Russian University. Wonderful talk. I really like that. Do you think it could be happening in other neurodevelopmental disorders also? Because, so what you're saying in terms of upregulation and downregulation is something that happened with, like, for example, the status epilepticus or seizure disorder, which is a prominent, and as you mentioned, they have that, many of the patients. And did you consider that seizure situation, the status, when you compare the control and the patients? And for example, one example of seizure disorders with immunological impact is MECP2 duplication. They are known for have immunological problem. Thank you.
So you're asking whether the seizure medications could have impacted what we see at the molecular level?
Not seizure medication, seizure situation, like seizure when they are active.
Right.
And they are not under control, which is very common in these patients.
Yes.
So they can trigger immune system, they can trigger different upregulation, downregulation.
Yeah.
And seizure per se, not like the other things.
It would be challenging to be able to unpick that, because when blood's drawn and when individuals have seizures won't necessarily be coordinated. But it would be good to look at those individuals that had more seizures, more frequent seizures, and the type of seizures, and to be able to correlate them with what we're seeing at the molecular level. I don't know if we could do that stratification with 22 individuals, but it would be a very good idea to look at that to see if it can inform what's happening when we compare the seizures to the molecular analysis.
Yeah, but, thank you very much. But I mean, seizure is one of them. For example, patients' weight, like nutritional status. Like some of them, they are not being at this age when they are young. You can't feed them through mouth, so they may have some alternative. So I mean, all of these factors, biological characteristics may affect or may not. I don't know.
Oh, I see. Well, I think I can answer your question in a different way. We're looking at blood biomarkers for other neurodevelopmental disorders, for example, like Rett syndrome. We haven't looked at MECP2 duplication, but we've looked at Rett syndrome, and we see a completely different response. These girls have seizures as well. They have neurodevelopmental delay, etc., but they have a completely different molecular profile to what we see with the STXs. So it is N equals 2. Different disorders, but they are very, very different profiles.
Yeah, thank you.
I was wondering if there is a microglial response that has been shown before. I'm not familiar with the literature that could mimic what you're seeing in the activated macrophage status.
Yeah, that's a really good question, and we've started to look at that because of GRN, which is highly expressed in microglia. So it could be something that's happening in the brain where the microglia are activated as well, and this is sending signals through to the periphery to say, hey, something's wrong here. But we haven't unpacked that yet. Yeah, but it'll be something good to have a look at, and what we're going to look at now is other genes that are also expressed in microglia or proteins to see if there's more of a correlation. Yeah. Thanks.